Right. So, continuing our inexorable march through the eukaryota, today we're actually going to end up covering the largest and most important of the single-celled eukaryote parasites. This is kind of the Reader's Digest version of a much fuller treatment that I give in the animal parasitology class, which I will be doing in summer one, hint, hint, if anybody feels like signing up for that. But let's go ahead and get started here. There's your classification scheme again. We've talked about all of these groups are based primarily on DNA analysis. We're leaving out a bunch of smaller and more obscure taxa that we just don't have time for. But we've mentioned the two huge supergroups, the Diaphoreticales and the Amorphia, as long as a third group, which may not be monophyletic. There is still debate about that. But we've covered just about all of the Diaphoreticales, with the exception of we're not quite finished with a subgroup of SAR, which is the alveolates. Among the alveolates, we've covered the ciliates and the dinoflagellates, and that leaves us the apicomplexans. These all have similarities in DNA sequence, but also remember that they've all got a common physical feature in that they've got this system of membrane sacs right underneath the plasma membrane. In the case of dinoflagellates, those membrane sacs are often filled with cellulose and they make up that sort of armor, the theca of a dinoflagellate. They do this in a small number of ciliates as well. They may have other functions in ciliates, but they're also present in apicomplexans, and that's why we group all of these as alveolates. So the ciliophora and the dinoflagellata, I've said my piece about. Apicomplexans we're going to see in lab on Friday, and I think unlike some other labs in this course, this one is just about guaranteed to actually work. Apicomplexans are called that because they all have a set of organelles at the anterior end of the cell, which is called the apical complex, which is a rather complex, I guess that's why they call it a complex, duh, a rather complex grouping of microtubules and other fibers, almost like a nose cone. And the apical complex is used to attach to host cells and in many cases to penetrate into host cells completely. Although the ones we're going to see on Friday do not penetrate into host cells. And they do this because all apicomplexans are parasitic. They may have fairly complicated life cycles. It depends on the species that you've got. But things like the malaria parasite has a complicated life cycle that alternates between a sexual stage in mosquitoes and rounds of asexual reproduction in a vertebrate host, notably people, and the ones that we're going to look at. Apicomplexans are all parasitic. They've got the apical complex, and most contain an organelle that looks like a chloroplast but isn't. It's called the apicoplast. Since apicomplexans spend most of their lives essentially in darkness, they don't photosynthesize. This is plausibly evidence that apicomplexans share a common ancestor with maybe photosynthetic dinoflagellates. They've retained this plastid, even though they no longer are photosynthetic. Presumably, they do something else with it. There does seem to be a lot of protein trafficking to and from the apicoplast. Some antiparasitic drugs that are being developed are targeting the apicoplast, which is a really promising line of attack because parasites contain the apicoplast, but their host cells don't. So if you could come up with a drug that would kill the malaria apicoplast, It would leave the host alone and presumably wouldn't have side effects. But I think we're still not entirely sure everything that the apicoplast is doing. Finally, there's never cilia or flagella. You may see some forms in this upcoming lab that can kind of glide. They'll just sort of very slowly glide from one place to another, and you won't be able to tell exactly how they're doing it. Others may be able to kind of contract and thrash back and forth a bit, probably because they've got microtubules under the plasma membrane, but they never swim. As I've said, they're all parasitic in animals. Human diseases include malaria, toxoplasmosis, which is not terribly dangerous for adult humans that get it, but it can be very dangerous to fetuses because if a pregnant woman gets it, toxoplasma can cross the placenta and do damage to a developing fetus. Cryptosporidium is another apicomplexan. There we go. One more joins our throng. Cryptosporidium is another one that you can get from contaminated drinking water. It's hard for water utilities to get rid of, and it's not terribly bad in most people. It'll cause you diarrhea, but most healthy people can fight it off, but it can be deadly in immunocompromised patients. Others that you'll encounter include things like coccidiosis. Your chickens can get that. That can go through your chicken flock like the reaper. Other coccidian species can infect dogs and cats. It's a common cause of diarrhea and even death in puppies and kittens, for example. Anybody going into veterinary practice is going to deal with a lot of coccidiosis. Babesiosis, aka red water fever, can infect dogs and can infect people. It can be lethal in cattle. It was eradicated in this country in 1948, but there's always a chance it might make a comeback, and it remains a problem in other countries. Okay, a lot of parasites may be anatomically simple, but the life cycle can get pretty complicated. There may be a lot of different stages that the organism goes through. The main parasitic stage is haploid. Most of the life cycle is spent haploid, but haploid cells will pair up and form gametocysts. And within the gametocyst, the haploid cells will divide into gametes. The gametes will pair up. Sexual reproduction takes place, producing zygotes. And the zygotes will usually immediately divide by meiosis to go back to haploid. So you do get alternation of generations from haploid to diploid, but the diploid stage doesn't last terribly long. The diploid stage usually divides right away to give rise to haploid cells. On top of this, a lot of species will alternate a sexual reproductive stage with asexual reproduction, which is called merigony. And if you might be familiar with this, any of you who took my evolution class this past fall, sexual reproduction has the great advantage that you're generating new combinations of genes. You generate genetic diversity through sexual reproduction. Stripped of all of the chocolate and the flowers and the Marvin Gaye records, sexual reproduction is genetic engineering. You're taking some of your DNA and blending it with somebody else's DNA to get new genotypes, new combinations, new shufflings of your deck, as it were. That can be advantageous, especially if you have a population that's exposed to challenges. The advantage of asexual reproduction is that you can do it much faster. You don't have to wait around for a partner. You can just start dividing and cranking out the offspring. However, the offspring are all going to be genetically identical to you, meaning if there's an environmental challenge like a disease or shifting conditions or a predator or something like that, all of your offspring are going to be equally vulnerable. And a lot of organisms are like Miley Cyrus. Um, I think this morning I'm not going to try to sing it, so I'll just say they get the best of both worlds, alternating between a sexual round and an asexual round. And you'll see that in the malaria parasites that I'm going to talk about in just a bit. So switching from sexual to asexual reproduction is something you see a lot. So about those features, the apical complex, there's a diagram of what it looks like in the human parasite Toxoplasma. One end of the cell is kind of pointed. That's the nose cone, as it were. And under the nose, there are a couple of polar rings. And within that, the conoid is made of these microfibers arranged in a spiral. So let me see if I can. Whoops, did not mean to do that. Let me summon up the laser pointer. So there's your conoid right there. You can see these microfibers are arranged in a spiral. And radiating from this, you have microtubules running right underneath the alveolar sacs. They're called subpellicular microtubules. Those run all the way along the body, and they're probably involved in locomotion. As I mentioned, apicomplexans can change shape to some extent, and they can glide over a surface, and they probably do it using those microtubules. So there's a transmission electron micrograph of what it looks like in Toxoplasma gondii. You can see up here and down there, you have those polar rings. Between the polar rings, you see this kind of diagonally crossed hatch area. That's the conoid. And then radiating down from here, you can see the subpellicular microtubules that would go all the way to the opposite end of the cell. So that's your attachment organ for latching on to host cells. There's Toxoplasma again. There's the whole thing. The entire parasite is in a vacuole inside a host cell. This is one of the ones that are intracellular parasites. So there you can see the apical complex. The conoid right there is labeled C. There are some filaments running back from the apical complex called micronemes. And back here you have these long sac-like organs that have very narrow necks that open up right here, kind of like really long vases that open up at the tip of the apical complex. Those are called rhoptries. And the micronemes and the rhoptries secrete proteins that allow the parasite to attach to and often penetrate into a host cell. And then back of that, you can see the nucleus back here. Um, these dark things are just labeled dark granules. My guess is they've got starch, but I forget what's in them. But the nucleus and nucleolus you can clearly see. So all of that apical complex apparatus with the micronemes and the rhoptries is there for attachment and penetration into host cells. Toxoplasma's also got an apicoplast, and this is that organelle that looks like a chloroplast but isn't one. And if you look at it carefully, you can see that it's actually surrounded not by two membranes, but by four. And I haven't talked about this that much. Maybe I should, but we think that the only uh, group of eukaryotes that established the original symbiosis between blue-green bacteria and the host that evolved into chloroplasts, we think that pretty much only happened once, and that was in the ancestor of the Archaeplastida, the green algae, red algae, land plants, and a few others like the glaucophytes. There are lots of other photosynthetic protists. We've seen dinoflagellates. Most of those are photosynthetic, and I've mentioned the brown algae and golden algae and things like that. Those descend from ancestors that either engulfed a green or a red alga and kept the chloroplast, or they engulfed a green or a red alga and kept the entire alga. So that's why this thing has four membranes. The innermost one would be the original membrane of the cyanobacterium. The second one out would be the membrane that encloses it in a green alga. The third would be the cell membrane of the original green alga that had it. And the fourth would be derived from the plasma membrane of the ancestor that engulfed the green alga. I'm not sure if I explained that terribly well. It'd be better if I could write this on the board. But let's just say that when you find plastids with more than two membranes, it's because either the plastids were taken from another species, that's called kleptoplasty, or they are descended from engulfing an entire alga and keeping the whole thing inside a cell. And this is what you see in dinoflagellates. Dinoflagellates got their chloroplasts by engulfing an ancient green or red alga, but not digesting the whole thing, just keeping it all there. The evolutionary history of chloroplast, if you map it out on a cladogram, it looks like the plot of a heist movie, where everybody is running around and shooting at each other and nobody quite remembers who's got the diamonds. Something like that. Apicoplasts contain their own genomes. They're involved in protein trafficking and fatty acid biosynthesis. And again, drugs are being designed that will target apicoplast function because apicomplexans have them, but their hosts don't. So if you can kill the apicoplast, you could hopefully kill the parasites and not touch the host at all. So there. The live group that we're going to see in lab are a pretty diverse group. They're called gregorines. They are parasites of invertebrates. The ones that we're going to see, I'm not entirely sure they really do that much damage to the host. They may be more commensal than truly parasitic. We know that all of their hosts have them. They're present in everybody, and they don't seem to harm the health terribly much. These are extracellular parasites. If you look carefully, you can see that on the anterior tip, they've got this weird little attachment organ that's transparent. You can see it enlarged right there and right there and right there. Those attachment organs will penetrate into cells lining the gut of a host. These happen to be dragonflies, and the rest of the cell will just kind of hang out. So that little anchor thing called the epimerite will penetrate into a host cell, but the rest of this thing will just hang out. The cells of this particular group of gregorines, not all of them do this, but the cells are actually divided by a septum into a front end called a protomerite and a back end called a deuteromerite. You can actually see the nucleus of that one right there. And they can get unusually large. A lot of a lot of apicomplexans are pretty small as these things go. But gregorines are nice and big. So you'll get to see these and ponder what they might actually be doing to their hosts. Apicomplexans in a group called the coccidia completely penetrate into host cells. Different species infect the intestines of vertebrates. The ones that people are most familiar with would be the ones that infect dogs, cats, and poultry. That is a stage of the life cycle that develops inside intestinal cells in chickens and then passes out in feces. So if you ever have to do a microscopic analysis of a chicken fecal sample, which we do in my parasitology course, it is possible that you'll see these. Come to think of it, you can also get these in sheep as well. I think there are other species that can infect other livestock. And this is the stage that will pass out in the feces, and it actually has to mature outside of a body for a little while, and then will pass to a new host if the new host eats anything that's contaminated with the feces of the infected host. I mentioned there are some coccidians that infect humans. Toxoplasma is one. This is the one that actually goes back and forth between an infective stage in mice and an infective stage in cats. And humans can get this if humans come into contact with cat feces, which is why my wife made me clean all the litter boxes when she was pregnant with our son. Cryptosporidium is another coccidian that infects humans, and again, it commonly comes in through drinking water. It's not lethal to most people, but it's one of the things that can be deadly to immunocompromised patients. I think it was the city of Milwaukee got cryptosporidium, a big outbreak in the drinking water back in the 80s. And there was a group called ACT UP, which was composed of HIV patients and their supporters lobbying for more attention to be paid to the disease. And they protested a lot because several dozen people with HIV were killed by cryptosporidium in the drinking water. There was a big outbreak, and it led to angry marches and protest activity. So it can infect people, and for some people, it can be quite serious. A group called the pyroplasms include something called babesia. That's cow blood, and pyroplasms, instead of being gut parasites, are blood parasites. They're spread by blood-sucking arthropods. Specifically, they're spread by ticks. So if an infected tick bites a cow or a rodent or occasionally a person, the bite injects a stage of babesia that penetrates into blood cells. Within the blood cells, right up there, I take it everybody can see my laser pointer. You can see several of these blood cells that have what look like little rings inside. That's the infective stage of babesia. And as babesia matures inside these blood cells, it begins to divide. And right over here, you have this blood cell with what looks like an X in it. That X is actually made up of four swarmer cells. This species, you have four, others, it might be just two. But this blood cell is going to burst, and these four swarmer cells are going to break out and go on to infect new blood cells. And so the cycle of infection can persist for quite a long time in an infected animal. Babesia causes red water fever in cattle, which was eradicated from the US, but not for a long time. It had been a problem ever since at least the 1840s, and I think it took till about 1940 to get it completely out of the United States. It's still a potential problem elsewhere. There is another species that can infect people that get bitten by ticks. It's not terribly common. It's not the worst thing that can happen to you if you get bitten by a tick, but it still is something that can cause symptoms that need treatment. And finally, Hemosporidia is the group that includes the malaria parasites. They also will alternate between a generation in a blood-sucking invertebrate, in this case, a mosquito, and they'll reproduce sexually inside the mosquito. And then they will be introduced into a vertebrate host, in this case, a human, by a mosquito bite, and they'll go through rounds of asexual reproduction in the vertebrate host. An infected mosquito will inject a stage called a trophozoite. Again, I'll never ask you to explain the meanings of these words, but tropho is a Greek root meaning to feed. So trophozoites are the feeding stage. They will get the trophozoites are injected and they penetrate into, I say vertebrate blood cells. Technically, in humans, the very first stage penetrates into liver cells, reproduces asexually in the liver, bursts out of the liver cells, and goes on to infect blood cells. So, in this human blood, over here on the left, you can see these are stained blood cells, and the blood cells are kind of pink. You have these blue things in here that this is actually called the wedding ring stage because they look like little diamond rings. The center of the ring is actually a big vacuole. The cytoplasm is all pushed out to the edge, and then the nucleus is the darker blue dot where the gemstone would be in the ring. So those are immature trophozoites penetrating into these blood cells and growing inside and breaking down their hemoglobin and eating it for lunch. And as they grow inside these red blood cells, they'll begin dividing asexually and they'll produce what's called a schizont over here. That is lots and lots of little swarmer cells called merozoites. When this thing bursts, the little merozoites are going to go spreading out and they're going to penetrate into more red blood cells and repeat this cycle. And a person with malaria infection can cycle back and forth between trophozoites. The trophozoites will infect blood cells. They'll develop into merozoites, which burst out. And that alternation, that asexual cycle, can go on for many years. If you and what makes malaria hell is that this cycle of developing and then bursting out of blood cells happens in sync. So if you're infected with malaria once every 3 days or once every 4 days, depending on what species you've got, merozoites are going to burst out of a whole bunch of your red blood cells in synchrony. Um, Byproducts of the ruptured blood cells get dumped into the bloodstream, and that triggers an immune response. So you get hit with chills. This gentleman, wrapped up in a blanket, is feeling terrible chills, even though he's in a tropical country. The chills get replaced by fever and splitting headaches. And then you get a period of sweating and exhaustion, and the whole thing repeats either once every 3 days or once every 4 days. So malaria is especially deadly when children get it. Most of the 500,000 people that die of malaria each year are children under 5. Older people may be able to live with malaria for many years again, but um, it's a serious drain on economic productivity. You can't work, you can't go to school, you can't do anything when you are flat on your back with the chills and the fever. So it's not only a killer, it's also a pretty serious economic drag. Recent interesting piece of news is that a company based in Africa has actually come up just a couple of years ago with a vaccine called Mosquirix um, that is not perfect. I think it only gives you about 30% protection. But in places where malaria is endemic, anything that lowers the risk is going to be helpful. 30% is a lot better than nothing. Oh, anyway, in people, a few merozoites in each round will transform into these gametocytes, this stage that floats around outside of the cells and looks kind of like a banana. That's the stage that infects a mosquito when the mosquito takes a blood meal. The gametocytes will pass into the mosquito. The gametocytes will transform into, they're called macrogametes and microgametes, but essentially they're eggs and sperm. Those will meet and those form zygotes. The zygotes form these cysts in the salivary glands of the mosquito and divide into millions of little trophozoites, which will get injected into a person that the mosquito bites again. And round goes the great circle of life. Sexual reproduction takes place in the mosquito. That gives you a new generation of infective trophozoites, which go on to infect a person when the person gets bitten. There's about five. There are five different species that cause human malaria, and as I mentioned, okay, in 2019 there were 247 million cases worldwide, killing over 600,000. Three-quarters of that being children under five. As I mentioned, that not only is there, you know, this is one of the great killers of the world, one of the great killer diseases that hasn't been conquered. Although Bill and Melinda Gates have devoted a very large fraction of their fortune to malaria eradication efforts. Probably the best things anyone has come up with is, in general, with parasites, often the most effective thing that you can do, and very often the cheapest thing that you can do, is try to block the life cycle. And so there are charities in places like Central Africa that distribute insecticide impregnated mosquito netting and teach people how to use that to keep themselves from getting bitten. And that's a very effective and a very low cost, low tech, locally adoptable measure that communities can adopt to lower the risk of malaria. That's a poster from World War II. Malaria affected our troops in the Pacific, killed about 60,000 U.S. soldiers fighting their way through the Pacific Islands, and was certainly a drag on combat effectiveness because even soldiers that didn't die can't fight effectively when they're lying flat on their butts in the hospital tent with the chills, fever, and sweating. So soldiers were periodically ordered to take these tablets that would protect them, containing a medication called atabrine, and they were ordered to use mosquito netting and make sure the mosquitoes didn't get in. So here you can see, don't go to bed with a malaria mosquito. That's probably quite a lot of things in that war that the soldiers had to be warned not to go to bed with, one of which was mosquitoes. Before intensive eradication efforts began, this is where malaria is or used to be endemic. In those darkest areas, you can see in Central Africa, those are areas where more than 75% of children between 2 and 10 had the parasite in their blood. And you can see we used to have this in the eastern United States, all the way up into Canada. Malaria could be a health risk. It used to be endemic in Arkansas. People used to get chills and fever and occasionally die from malaria infection. Even here, last I looked, we had not completely eradicated it, but we've made it extremely rare. But it used to be not so rare in the South and Eastern United States. Oh, by the way, the first really effective treatment for malaria was an extract from a South American bush called the cinchona, and an extract of the bark of that bush contained a compound called quinine. Quinine was, it still got some nasty side effects, but it was the first really effective anti-malarial treatment. And when the British colonized India, you can see India malaria is endemic. There's pretty high rates of infection. The first guy to piece together, okay, mostly the first guy, there's politics involved, but the guy who got the credit for piecing together the malaria life cycle was a man named Ronald Ross, and he did his research in India. So malaria was a serious problem there, and he worked it out while doing research on malaria patients in India. He was British. And even before the life cycle was worked out, we had this quinine, and British soldiers in India were issued a daily dose of quinine just as a prophylactic measure to keep them from getting malaria. The problem is that quinine is exceptionally bitter. It tastes really nasty. So somebody got the idea that it would go down much more easily if you added lime juice and gin. That's the origin of the gin and tonic. The gin and tonic originated as a way to make drinking your anti-malaria drugs just a little bit less nasty. Tonic water, I mean, you can buy this in the grocery store to this day, and it still has quinine, but modern tonic water only has a tiny bit to add bitter flavor. It's not strong enough to protect you from the malaria parasite. It's not strong enough to give you side effects. It's not nearly as strong as the quinine that British troops in India were taking. But yes, the gin and tonic, my grandmother's favorite cocktail, um, owed its existence to anti-malaria efforts in colonial British India. So now you know. That's all I'm going to say about Apicomplexa. There's this group that may not be monophyletic. There is still some question about that. The status isn't completely clear. I'm going to treat it as one group with a little asterisk by its name. It's called the Excavata, and the Excavata contain most of the other potentially dangerous protist parasites of humans and other animals. Unlike apicomplexans, most excavates have flagella. They get their name because many, although not all, have a specialized food groove, a little pit or groove or something like that in the cell that enables them to feed. A lot of them have lost their mitochondria, but they retain DNA sequences that were transferred from mitochondria in the past. So even the ones that don't have functioning mitochondria now are descended from ancestors that used to have them. Some excavates retain organelles that are probably highly modified mitochondria, things called mitosomes and hydrogenosomes. There's a fair number of free living excavates. We could probably find them in Lake Beaver Fork, or at least we will when it gets a little warmer and sunnier. Today is not a good day to go to Lake Beaver Fork or, frankly, anywhere else. And we'll see a couple of those free living ones. But there's also a lot of pathogens and symbionts in the excavata as well. So one major group of excavates is called the metamonads, and these do seem to be monophyletic. And there's a fairly typical metamonad that is a symbiont in humans called Trichomonas vaginalis. As you can tell from the name, it does in fact live in the human vagina. Um, a long time ago in a galaxy far, far away, I was a volunteer medical lab tech at a place called the Berkeley Free Clinic and looked at a whole lot of samples. I've seen an amazing amount of urine in my time and can tell you all sorts of things about yourself from doing urine tests. But we'd also see vaginal samples from women, and every now and then we'd find this. And sometimes you'd find a few in women that weren't particularly ill. So in low levels, it doesn't always cause problems. High levels of Trichomonas vaginalis can cause soreness and itching and a distinctive odor and discharge. It's not lethal at all. It's just kind of miserable to have trichomonas infection of the vagina. So I used to see this under the microscope on a fairly regular basis. They were kind of entertaining to watch because they've got four flagella. You can see three of them sticking out of the anterior end of the cell, but the fourth one is this curlicued thing. By the way, this picture's been artificially colored. The real ones don't look orange and yellow, but this thing that kind of does a sine wave back and forth is a fourth flagellum that is tethered to the cell body by a fold of the cell membrane. It's called an undulating membrane, and it's kind of like a long fin. And when the organism beats that fourth flagellum, instead of just thrashing back and forth like a whip, it makes that fin ripple. Kind of hard for me to demonstrate how it happens, but I used to see it all the time. There also happens to be a rod-like structure that runs through the entire cell, and that's the end of it protruding out of the bottom. That's called the axostyle. You can find Trichomonas, other species, in the guts of termites. There's a lot of this particular subgroup of excavates that are gut symbionts in termite guts. This is one we used to see a lot. Some years we do this here and some years we don't. I'm not really sure if you've done this or not, but some years in 1440 or maybe 41, we order termites and squeeze them until they release a little drop of gut fluid. And the gut fluid contains samples of the symbiotic bacteria and protists that live in termite guts and make it possible for them to break down wood. And I used to see Trichomonas in termite guts, and you would see a lot of Trichonympha as well. Trichonympha has lots of flagella. You can kind of see them as all this shaggy coating right there. Those are all long flagella. It's got this kind of pear shape, but as the flagella beat, it can change its shape quite a bit. It can even do this kind of wobble. It kind of shakes when it laughs like a bowl full of jelly, right? Kind of like Santa Claus. Um, and Trichonympha is also packed full of symbiotic bacteria. And in this posterior half of the cell, these dark, irregular, grainy things right there are tiny fragments of wood. And the bacteria in the Trichonympha digest the wood. So the termite survives because it contains Trichonympha, which contains bacteria, which break down the wood. So we've got symbionts nested one inside the other, like so many Russian dolls, which I happen to think is quite nifty. This is another termite symbiont, Caducea, which I don't think I've ever seen. It may not be found in North American termites. And I mentioned this to give the cell biologist who might be listening to this something to think about. The outside of Caducea is covered in bacterial symbionts. You can see them, they've stained in green and red. So you have a light micrograph up here at the upper left, and then at the upper right, you have all of these green dots. You have those red dots right there, and then at the lower right, that's a composite of all three pictures. Those dots are bacteria that cover the outside, and it's the beating of the bacterial flagella that enables Caducea to swim. It would be sort of like making a suit and gluing millions of flies to the outside of your suit, and then getting all of those flies to beat their wings at the same time and flying away because you're covered in a fly suit. I don't think that would really work, but that's essentially what caducea is doing. It's got these trapped bacteria on the outside, and their swimming is what propels the entire organism forward. And the reason this might matter to cell biologists, the reason this might matter to cell biologists is that as caduceus swims, the anterior part of the cell perpetually rotates around the central stalk called the axostyle that supports it. And that was a key piece of direct evidence back in 1976 for the fluid mosaic model of cell membranes. I know you know this, that the cell membrane is a lipid bilayer, right? It's a layer of, or a double layer of phospholipids with the hydrophilic heads sticking out and the hydrophobic tails sticking in. Well, that means that the cell membrane is more of a two-dimensional liquid film than it is a solid. The cell membrane is not like, the rubber of a balloon. Um, and this is how we know that. If you take a balloon and you take part of the balloon and you rotate it with respect to the other part of the balloon, you're going to twist that balloon up into two bubbles. If you've ever seen somebody making balloon animals, this is how they do it. They take part of the balloon and twist it and pinch off these little bubbles that they then bend around into the shape of a poodle or whatever it is that the balloon guy is making for you. This doesn't happen. Caducea can have its nose cone rotating without ever twisting itself into two. And the explanation is that the cell membrane must be a two-dimensional fluid instead of being a flexible solid like rubber. So this was actually historically a key piece of evidence for that fluid mosaic model of cell membranes. And kind of one of my great messages of this course, we cover so many different critters, and it's hard to come up with overarching principles. But one thing I do want to make sure I make a point of is now and again, studying the really weird and obscure stuff can pay off really big. There's more than one case where we've made really major discoveries, but not by looking where you might expect, looking at unexpected organisms in unexpected places where most people might not go. Uh, now and again, now and again, you find all kinds of things in weird places. And by the way, just for the record, I have just now gotten an update from UCA, and we are continuing remote instruction tomorrow, Wednesday, January 28th. They are doing essential services only on campus. Lectures are going to be done by remote. Essential services and personnel only on campus. We're not going to have a lecture this coming, tomorrow, but your other classes are going to be virtual or, otherwise not held as usual. So I just now got that update as I was talking about Caducea versatilis. Right. A group called the Fornicata, tee hee hee, includes Giardia. Giardia is extremely small, but Giardia can grow in huge numbers and attach to the cells of your intestinal lining. And when it does that, it blocks your ability to absorb things through your large intestine, like water and fat. And the result of Giardia infection is a rather miserable condition called hiker's diarrhea. It's called that because it's spread through feces and it can live in the guts of quite a lot of mammals. So if you're drinking water that's been contaminated by animal feces, even in wilderness areas, um, you can ingest Giardia cysts. One another name for Giardia infection in humans, by the way, is beaver fever, because it can survive in beaver guts and pass out in beaver feces, and then get drunk by people if you who drink water from a stream where beavers might be living. Even pristine, clear running mountain streams that look like something out of a Coors beer ad can potentially be contaminated with Giardia, because we all know what bears do in the woods, and bears can transmit Giardia. This is why you've got to boil or filter or chemically treat um, any source of water that you get from the wilderness. If you're going on a hike, it's dangerous to drink unfiltered, water from rivers or streams or lakes or anything like that. And if you're a hiker, you need to have some sort of water purification kit in your backpack, or you'll get massive infection from Giardia lamblia, which again is not usually fatal, but it's pretty damn miserable. The word I hear describing hiker's diarrhea, the word that gets used a lot is foamy. So you do not want foamy diarrhea when you're on a hike. So that's a piece of intestinal epithelium. And every one of these little round, kind of turtle shell shaped things is a Giardia cell that has latched onto the intestinal epithelium and causes malabsorption syndrome. And that just means that the intestine can no longer absorb what it's supposed to absorb. You see this in a lot of different diseases. And the result is that since you can't absorb water and nutrients through your intestine, they just pass on out of the body and you get diarrhea. Giardia can reproduce extremely rapidly. A severely infected person can pass 14 billion Giardias in each stool. So there, you don't want to get this. There's a second excavate group. The ones that we've seen are called metamonads. The other one is called the Discoba. And they include some free living species, including some that we can see in pond water from around here. A group called the euglenids. They're free living excavates whose ancestors engulfed green algae and picked up their chloroplasts. This is one of the more common Euglena species. It's green and it's photosynthetic. It has a flagellum coming out right here. I think actually it's got two that it uses to swim, and it is capable. There's a little mouth here at this end that it can expand and engulf smaller food particles. So Euglena can feed as well as photosynthesize. Best of both worlds again. Over here on the right, Paranema is very similar to Euglena, but it does not photosynthesize. Paranema is fairly common in biology labs because it happens to have, whoopsie, sorry about that, because it happens to have a very prominent flagellum that's really easy to spot. So right up there, let me get back my laser pointer. You can see the flagellum really well. Euglenids are related to a group called kinetoplastids, and some of these are also free living. But kinetoplastids also include a lot of parasites that get spread by biting flies. On the left, you have trypanosoma. That again is human blood, and these kind of curly things here that are wriggling around among the blood cells. There's one up there at the left. There's one right there. You can see a prominent nucleus in the middle. Um, Trypanosoma brucei causes a disease called African sleeping sickness. which mercifully has gotten a lot more rare in recent years now that eradication efforts are seriously underway. But African sleeping sickness will get in the blood, will also get into your cerebrospinal fluid and cause confusion, lethargy, and disruption of the sleeping cycle, and ultimately death. Trypanosoma cruzi from South America, you get from the bite not of a fly, but of a blood sucking true bug called triatoma. And that causes a chronic disease called Chagas disease. On the right, that's leishmania, and that's spread by biting sandflies. That can cause various diseases, sometimes of the skin. You can get this eruption of lumps on the skin that used to be called Jericho buttons because this used to be endemic to Israel and Palestine. You can get very severe disease of the internal organs called kala azar, and this tends to be prominent in the developing world in Central and Eastern Asia and the Middle East. It happens to be spread by the bite of sandflies. There have recently been a few reports of dogs getting this in the United States. So in the US, we're not entirely immune from leishmania infections. What gives these their name is a structure called the kinetoplast. So there's this would be Trypanosoma cruzi. There's a single flagellum, and like Trichomonas, the flagellum is actually attached to the length of the cell body by a fold of membrane. So that flagellum forms a fin. You can kind of see it, this thing trailing back there, that's the flagellum. And if you look carefully, you can see that fin that attaches the whole thing. That dark spot in the center is the nucleus. And then right up here, that is the spot where the flagellum attaches to the cell body. It's a dark stained little spot called the kinetoplast. So right there is a kinetoplast. There's a kinetoplast. Right, the nucleus is farther back, and then there's the kinetoplast. There's the nucleus, and there's the kinetoplast. There's the nucleus, and there's the kinetoplast, and so on. And it turns out that the kinetoplast is made up of a whole bunch of interlocked circular pieces of mitochondrial DNA. There is a large mitochondrion uh, right where that flagellum attaches, and the kinetoplast is a big lump of mitochondrial DNA. You've got dozens of coding circles of DNA called maxicircles, and thousands of much smaller circles called minicircles. And it's all right there. So we will look at some prepared slides of this in Friday's lab, and it just might interest you to know that what you're looking at is actually mitochondrial DNA. You can't clearly see the whole mitochondrion, but you can see under staining the mitochondrial DNA. Just a little reminder that mitochondria are descended from free living bacteria that used to have complete genomes of their own. Mitochondria still have a bit of DNA, and this is one of the few cases where you can see it. So there. And there you can see how that flagellum forms that undulating membrane. Different trypanosome species and different kinetoplast species may or may not have that membrane. They can have different arrangements of the flagellum. We don't really need to get into that here. Last group I'll talk about, the Discoba, includes those euglenids, the kinetoplastids, and the heterolobosians, which are near and dear to my heart because I did my undergraduate honors theses on heterolobosians back all the way back in 1991. A long, long time ago in a galaxy far, far away when hair metal still ruled the music charts. And I mentioned earlier that amoebas are actually polyphyletic, that um, habit of having no fixed shape and just the ability to just kind of crawl and ooze everywhere, that has evolved independently in many different lineages of protist. So most of what we call amoebas are not closely related to these guys. The heterolobosia includes amoeboflagellates that can actually switch back and forth between amoeba forms and swimming forms. So right up here, that is a little free swimming form with a couple of flagella, and this will swim around in the water and do its thing. And when it finds a place to settle, it will suddenly change shape. It'll lose that streamlined shape and begin looking blobby. And those flagella get shorter and eventually disappear, and the thing will transform into an amoeba. Um, Many species actually have a third stage. The amoeba can transform into a resting stage called a cyst. If in soil, for example, if soil dries out, these are very common protists in soil. That's what I actually studied when I did my undergrad thesis. And they can dry out, build a kind of cellulose wall around themselves, and just go into suspended animation for months at a time, and then come back to life if the water returns. So these are kind of neat because they can transform from a cyst stage to an amoeba stage to a flagellate stage and back. The one that I studied back in the day was called Nigleria gruberi, and gruberi is very common in soils. It turned out to be very common around plant roots, where it's a major player in this very specialized ecosystem of microbes that grow in the soil immediately surrounding the root of a plant. Little ecosystem called the rhizosphere. And Naegleria gruberi is harmless and ultimately probably promotes plant growth and fertility just because it's part of this ecosystem. Naegleria fowleri is very much like Naegleria gruberi, except it grows at higher temperatures. You're not going to find it in your typical soil. You can find it in bodies of water that are unusually warm. If they're very shallow in warm climates, you'll get them. And the problem with Naegleria fowleri is that it grows very well at human body temperature. And what Naegleria fowleri can do is usually crawl up your nose if you go swimming in water that's infected with Naegleria fowleri. Even then, you don't always get it. This is actually pretty rare. But if you inhale some water up into your nose, maybe by, cannonballing off the dock without wearing nose plugs or something like that, and you get this water deep up into your nose, Naegleria fowleri can crawl right up your olfactory nerve into your brain and start eating your brain. These are the so-called brain-eating amoebas. It is extremely rare, but it's almost always fatal because symptoms start out as being kind of flu-like symptoms, and then it starts feeling like meningitis. And by the time the doctors can diagnose it, it's usually already too late. You have to give huge doses of drugs to try to kill it, and most of the time, those don't work. This is not something that most people really have to deal with. Your average swimming pool is going to be too cold and also too chlorinated to get these, so go swimming at the aquatic center all you want. And even if you swim in, infected waters like, Mima's old swimming hole or something like that, you're not likely to get it. If you're paranoid about getting it, a simple nose clip will save you from getting it. So we get usually it is tracked by the Center for Disease Control. We usually get maybe 2 to 3 cases every year, but it tends to get announced on the news Um, and I've seen people who are a little bit more scared of it than they probably have to be. And we did have a couple of famous cases around here. We used to have this water park in South Little Rock called Willow Springs that was very proud of their all natural water that was untreated and unchlorinated. And in something like '07, a kid actually died from Naegleria that they'd caught at Willow Springs Water Park. Shallow water in summer gets warm enough that it can sustain Naegleria fowleri. And then it happened again in July 2013. This girl, Kaylee Hardig, went swimming at Willow Springs and got Naegleria. And she made history by being something like the third person in 50 years that survived this. They took her to Arkansas Children's Hospital. They were able to get a experimental drug. They had to get it from Germany because it had not yet been approved in the U.S. They treated her. She survived. She regained consciousness. She did have some deficits when she came back because the amoebas had been eating her brain. I'm told that those mostly went away in time, and she is now, I mean, I guess she'd be older than you folks. She'd now be about 25. And as far as I know, last I heard was, living her best life. By the way, this is why Willow Springs Water Park has closed. I don't know if any of you ever went there, but you can't go there now. Remember, we have like 3 cases of this a year, and then for 2 cases in 5 years to pop up at the same place, well, that's a problem. And that is why Willow Springs Water Park is no more. All right. We seem to have ended just a little bit early, and I'm not seeing any. Let me just check the chat. I'm not seeing anything new on here. So I'm going to go ahead and stop the recording.